Hello, guys. Welcome to the first skill building session of season 6. I am Sunipun. I'll be your TA for skill building. A bit about me. I am a senior in high school. I will be going to MIT this fall, and I was a finalist last year, and I was a bronze medalist. I will be teaching you how to read papers mostly, but along with that, I'll be teaching you various things. I guess so. USABO currently is a bunch of I don't know how usable questions are going to be in the near future, but I do know Part B will still have questions based on papers because that's what Part B is kind of for. As such, if you know how to read papers and if you know how to read figures, you'll do really great on usable, which is why I am doing this office hours. So in order to read papers, you need to first understand, I guess, the components of a paper and what the experiments in each figure do. And that is not something that's particularly difficult to do, but you need to practice it. And over time, it is something that you develop. What we will do in this class is that we will read through a lot of papers. And before each paper that will consist of information that you don't know, I will do a primer. For example, if there's a genetics paper with lots of techniques or techniques like Hi-C or immunoprecipitation or ChIP-seq that you're unfamiliar with, I will do a primer the day before and then the week before, and then we will have our paper discussion. Usually I will send out a paper the day I do the primer, and then you will read the paper and then we will discuss it. There is a slideshow for this, which I will post after the class. Yeah, it is. But anyways, I will post a slideshow after the class. How we will discuss the paper is that we will specifically look at the figures in each paper. The crux of every paper are the figures within it. So we will evaluate the paper by looking at the figures. And today what I plan to do is that I want to take people through the process of answering a question in science. Some may call it going through chapter 1 of Campbell Bio, but I'll be doing that, except I will just do an example of it. So I will start screen sharing and then I will get started. Can everyone see the screen? I hope everyone can see the screen. Okay. First, yes, please stop replying. Do not remove Kevin Bao. Wait, which Kevin? Ajay, you are the guard. You manage this. I gave you co-host. Just fix everything. Do not ask me to remove anyone. Ask Ajay. Anyways, we will get started. So first, I will teach you guys about what forward and reverse genetics is. While many people think of forward and reverse genetics as mainly Drosophila work, it is actually used in everything. A lot of research of mostly, so if you read a book called MBOK, which is a really, really good book, you will see the names of lots of genes that don't make sense. Most of the time, the genes that you will see are probably from the fact that they're probably from a genetic screen. A lot of times, they're from forward genetic screens. In some ways, forward genetic screens are a bit harder than reverse. But that's also not a good statement to make. But yeah, so as you can see, there are so many methods listed here. We are not going to go through all these methods today. Some of the methods we will go through in the class include genetic mapping. We'll go through QTL-seq. I don't know whether we'll go through BSA. We'll go through transposon RNAi, CRISPR, and some other techniques that are not listed here. But we will go through processes of how this happens. So a forward genetics is when you have a variation or a mutant. You basically have a mutant phenotype. So it can be a fly that cannot sleep. Who asked me? Wait, Sunipun, is today just learning how to read papers and stuff? Today is me giving you an example of a scientific discovery. You didn't get it? Wait, what? Okay. Today, okay, we will learn how to read papers from next week. That's what next week is for. Today is to kind of show you how science works. Because if you haven't worked in a lab, it's unlikely that you would have been exposed to the process of scientific research. And if you don't know how that process works, then it's difficult to think about figures and like you don't know how to think about it. For that, I think a basic knowledge of how a research process works is important. And so I will go through an example of it. And as you go through the example, I will point out certain things that I notice, and I hope you will notice those too in the paper that you read for next week. So forward genetics is when you have a mutated phenotype. So often in forward genetics, what you do is that you take a fly, you mutate it using X-rays, and the X-rays mutate in random places in the DNA, and then usually sometimes those are recessive mutations. So you take two mutant flies, you cross them together, and then among the flies that you do get, you will see some 1/4 of them, because it's recessive, 1/4 of them will carry a specific mutation. And from that mutant fly, you can get to the gene that is causing that mutation or is implicated in that phenotype. That is what forward genetics is. Reverse genetics, as it probably sounds, is the reverse. You have a gene and you want to know what that gene does. In forward genetics, you have a mutated animal and you want to know what mutation in what gene is causing that. So today we will go through an example of forward genetics. So in forward genetics, the first step is always a mutant animal, something that is mutated. That's the starting point. If you have a mutated animal, then you can learn more about what is mutated in that animal, how it got mutated, and so on. And then from that, you can go on to the gene that is responsible for it. I only have 12 slides for today. However, each of the slides I do believe are quite dense. So I will advise everyone not to look at the whole slide, but to just look at what I am looking at with me. So let's look at this. This is a mouse. This is a mouse leg. This is another mouse leg. As you can see, it is similar to humans. I guess, yeah, it's paw, never mind. But like, it is very similar to humans. This is the thumb. You can classify this as 1, 2, 3, 4, 5. 1 is the thumb, 2 is the index, 3 is the middle, 4 is the ring, and 5 is the pinky finger. But we have a mutated phenotype. This mutated phenotype is called hemimelic extratose. As you would probably see, there are extra toes here. We normally have the digits that we're supposed to have, and there are extra digits. So this is definitely a mutated phenotype. Now, you can do forward genetics on this. You can try figuring out the mutation in what gene is causing the mouse to have a duplication in its digits. These are the digits to normally have. This is 4. No, this is not 4. This is 5, 4, 3, 2, 1. And then we have another 2, 3, and 4. These are abnormal digits, so I guess they don't really follow the 2, 3, 4 convention. But we do see a duplication of digits along this axis. Now let's learn about how our limb axis is like, how is it arranged? So this part of our arm, there's a bone. There's one single bone here that's called the humerus. This part of the arm has two bones. The upper bone is usually the ulna, and the upper bone is the radius, and the lower bone is the ulna. So if you wanted to assign directions to this, you would put our thumb is in the anterior position, and our pinky is in the posterior position. So is the radius anterior or posterior to the ulna? Also, I will give about anterior. Yes, that is true. The radius is anterior to the ulna. And you see digit 2 is anterior to digit 3, and digit 4 is posterior to digit 3. It's important to understand how these axes work before we go into how these axes are generated in like animals. So also, these are not metacarpals. These are carpals, but like the book labeled them incorrectly for some reason. More finger is not necessarily good evolution on everything. But yeah, why is it not using ventral dorsal? So ventral dorsal is a bit different. This is dorsal, this is ventral. This is a chick embryo. This is a chick limb. Chick is a very common model for understanding limb development. Our mutation is in mice, but in chick, it's also very similar. The process of limb development in chickens and mice, Sonic Hedgehog is indeed the problem here, but we will look at it. Chickens and mice have a very similar process of limb development. So we are looking at the chick in order to understand the axis. Our thumb is the anterior. Why did Udi ping me? Udi, what do I need to know? I'll just keep going. And our pinky is in the posterior. But yes, these are the axes. So I think you'll agree with me if I say that this irregularity is not an irregularity in the dorsal ventral or the proximal distal axis. It's an irregularity in the anterior posterior axis of the limb development. And this abnormality causes a digit duplication. It could cause deletion and a lot of other abnormality. But in this case, it causes a digit duplication. And we want to find out why this happens or what gene might be implicated in it. This basically says what I previously said. The thumb is anterior, the pinky is posterior. And the hemimelic extratose is the mutation that this is officially called. I will call it HX for short. And it's a mutation in the anterior posterior patterning because the anterior posterior is the organization of our thumb to our pinky. And we see a duplication of the digits. Digits basically mean fingers. Does anyone have any questions so far? I will address those before I move on to the next slide. Questions relevant to this slide, not questions that are far beyond what this slide covers. I should also check Q&A. How do I check Q&A? How do you know that the fly gets a recessive mutation? Is there a way to guarantee it? There is a way. There are techniques that Drosophila geneticists use in order to deal with whatever type of mutation you have. I don't know much about it, but there is a technique that I know called fused X chromosomes, which can help you. So since if the mutation is recessive, you would have to go through more generations of breeding in order to actually get it out in like the offspring. Fused X chromosomes are a technique in which you can do it earlier. However, that is a content for another class. I plan to cover how we do that in a different class during genetics. So we will discuss that then. But yeah, it can be both dominant and recessive. Like we cannot guarantee whether it's anterior dominant or wait, is SHH mutations that also cause holoprosencephaly? It can, yes, because SHH is very important for closure of the neural tube. It can cause anencephaly, and if it's in certain cases, it can cause holoprosencephaly too. Also, I wanted to mention this. You might have heard about the terms anencephaly and spina bifida. So we have a neural tube inside our body. Not right now, but when we were an embryo, we had it. That neural tube has to close at the anterior portion. For us, our thumb is anterior and our finger is posterior, but our head is also anterior to our legs because anterior posterior axis is top and bottom basically. So our anterior neuropore that leads to our head has to close, and our posterior neuropore has to close. If our anterior neuropore doesn't close, then we have anencephaly. If our posterior neuropore doesn't close, we have spina bifida, and that can be spina bifida cystica or spina bifida occulta. So we don't need to know that, but there are lots of diseases that can happen with neural tubes, and that's not relevant, but that's just a fun fact, I guess. It's all because of Evan that it went to this entire description. It's all Evan's fault. Anyways, we will move on. So this is a limb bud. What is a limb bud? A limb bud is exactly what it sounds like. An embryo usually looks like this. This is like an embryo. It has this notochord, and behind the notochord, it has its dorsal hollow nerve cord. Dorsal hollow nerve cord basically tells you that it's dorsal to the notochord itself. And at a later stage in development, you see the development of buds. In the case of chicks, these are wing buds, and these are called leg buds. So what do? They're just buds And these buds are a mass of proliferating cells. These mass of proliferating cells extend outwards and form like the actual wing or the leg structure. That's what these buds do. Now, along these buds, we have different regions of patterning. So this region is called proximal. Proximal basically means closer to our arm. So like closer to like a shoulder is proximal. If it's closer to like our wrist, then it's more distal. There's proximal distal patterning, there's ventral dorsal patterning, and there's anterior posterior patterning. We today are exclusively looking at anterior posterior patterning. So if you think about it, this top part is the anterior part of the limb bud. Yes, it is. Jason is right. And this part is the posterior part of the limb bud. A long time ago, a scientist named John Saunders. So John Saunders was a neurologist. He was not a developmental biologist. He accidentally took a part of the posterior section. It was not called ZPA then. He took a part of the posterior section of the limb bud and transplanted it to the anterior section of another limb bud. And guess what we found? We found a digit duplication very similar to the one that we now see in hemimelic extratose mice. We have digits 2, 3, 4, and then we have another duplication of digits 2, 3, 4. This would be the humerus. This would be the radius. This would be the ulna. This would be the carpals. And this would be the metacarpals. This is anterior and this is posterior. Again, this is who said something in the chat? Joseph said something. It's 4, 3, 2 that's duplicated instead of 1, 2. How do we tell? Because the metacarpals are different. You can look at the structure of the metacarpals and determine what digit it is because of anatomy alone. So this is the characteristic of how 2 looks. You see that there's no 1 here. That's because the 1 is kind of fused at like here and it doesn't really get space to grow. There's no 1 here, but through anatomy, you can look at the finger. If you go to a doctor, a good doctor, and if you show them like a bone of like any part of your finger, they should theoretically be able to recognize what finger it is from. In theory, they should, in theory, be able to, because I think they do that in med school. And if they didn't, then I guess don't get sick. But yeah. So one of the themes that we need to know is that whenever we have a problem in science, we usually try looking at similar problems that people in the past have struggled through. The scientists who had the problem of the hemimelic extratose mice with the duplicated digits looked at previous problems where we also had duplicated digits. They found this example where John Saunders transplanted the posterior section of the limb bud and transplanted it into the anterior section, and we found a digit duplication. This posterior section of the limb bud was later called ZPA. Z stands for zone of polarizing activity. Now, how do you interpret this experiment? What is the ZPA doing? There are two hypotheses that the ZPA, there are two hypotheses like regarding the role of ZPA. ZPA is a zone of polarizing activity. It is the posterior portion of the limb bud. So in this case, it was not called ZPA during the time of this experiment. They just took the posterior portion of a limb bud. This is a limb bud, by the way. They took the posterior portion and they transplanted it into or grafted it into like the anterior portion of another limb bud. That the posterior section of a limb bud is the ZPA. You can think of it like that, and that will be good enough for the purposes of today. So what is the ZPA doing in order to cause this? So if we think mechanistically, the ZPA can do two things. I will first erase this so that there's more space first. We know that the ZPA is forming these extra 3 digits somehow. So we can say that the ZPA is physically, the cells of the ZPA, or the cells of the posterior section of the limb bud, is physically forming digits 2, 3, 4. These extra 2, 3, 4 digits are what causes a digit duplication. And the ZPA physically forms these 3 digits. That's one hypothesis. The second hypothesis is that the ZPA does not physically form the three digits. It induces the cells around it. So this is the ZPA. This is the ZPA. It's been transplanted here. These are the cells around it. The second hypothesis is that the ZPA does not form these cells. It induces, it sends out molecules that induces the cells to form digits 2, 3, 4. What is the difference between these two hypotheses? In the first hypothesis, you are claiming that the ZPA itself is forming the three digits. In the second hypothesis, you're claiming that the ZPA is inducing other cells to form those three digits. These are very distinct hypotheses, and mechanistically, they matter a lot. However, now that you have come up with this hypothesis, how can you what hypothesis in theory couldn't another hypothesis be that ZPA suppresses other cells to form the digits? How would that work? If the ZPA suppresses other cells, then why are you putting the ZPA somewhere else? Cause duplication. It probably wouldn't. So if the ZPA were to suppress limb formation, then we would see like not limb duplication, limb deletion when we craft the ZPA. So that is not the best hypothesis. What if it suppresses cells in their inventory? That can happen. However, we have something called Occam's razor or like parsimony. We try to go for this explanation that makes most sense easily. What's the question? We picked the simplest one. Usually, yes. Explain why my thing wasn't correct. Because the ZPA normally is in the posterior section of the limb bud. Now, we have normal development when it's in the posterior section. But if you graft it to the anterior section, we get duplication. If it was suppressing the formation of digits, then if you have two ZPAs, one on the anterior and one on the posterior portion, you would get nothing or you'd get deletions. You would not get duplications. That is a skill that you should have. You choose a hypothesis and then you check whether that works by looking at what would happen if the hypotheses were true and whether that matches with the experimental results. So we have two different hypotheses here. There's one way you can check which hypothesis works. Would anyone like to try? Would anyone like to propose a way you can check whether hypothesis 1 or hypothesis 2 is true? This is probably hard, so I'll give 10 Bao bucks to whoever. Senior Kupeling Chi. Yes, you can unmute and tell me. 10 Bao bucks to whoever gets it completely
Okay,
correct.
wait, so whoever can I assume that if you want to test wait, are we allowed to use any tech or is it like
You can use anything except sequencing.
except sequencing? Oh, okay, wait, then I'd assume that just like cause a knockout of ZPA, right? And then cause a knockout or knockdown of ZPA and then graft it and then if it doesn't form it. Wait, hold up. Let me think real quick.
So something that you should think about is how the result of the experiment differ if hypothesis 1 versus hypothesis 2 were true. There is a much simpler way to do this. But since you chose knockout, you can use that.
Wait,
Hy
if it's like
hypothesis,
sorry,
yeah,
if it's
sorry, go
like
on.
physical formation, can't you just graft it to like a non-limb bud? So then like it'll form anyway, but if it's like using the molecules, then if it's in a non-limb bud, then it can't form because like the things that form it like aren't actually there.
Good question. Good point. So there is a concept of necessity and sufficiency. So necessity means that you need something if let's say digit formation is ZPA is necessary for digit formation. That means you need the ZPA for digit formation. If you knock out the ZPA, digit formation will never happen. That's what necessity means. Sufficiency means if you put the ZPA in another ectopic location, that alone will be enough to induce the formation of the digits. However, even if you take out the ZPA, the digit formation can still happen. It's not necessary, but it is sufficient in order to form the digits. What you're saying is a test of necessity and sufficiency. If you put the ZPA in another location and you see that the ZPA is leading to the formation of digits, then you say that it's sufficient for the formation of digits. However, that is not what we're looking here, because an argument to your question would be, let's say that it can only induce specific type of cells that are in the limb bud, because limb bud has a specific type of cells that are not found like everywhere else in the embryo.
Yeah.
So
Sorry.
maybe
Wait, because then
yes.
like if you transplant it and it still forms
Mm-hmm.
the like things, then like wouldn't that support like physical, because like it's straight up just
Maybe it
doing
needs something,
mitosis.
maybe it needs a factor in the limb bud and itself. Maybe outside the limb bud, it cannot, maybe it physically still forms the digits. But if you take it outside the limb bud, it cannot form it because it doesn't have the right signals to form the ZPA. Because like the limb bud is a very special environment. And what you're saying is correct, but like that's a test of necessity and sufficiency. That doesn't help you determine, because even if hypothesis one is true, you can still, even if hypothesis one is true, it wouldn't necessarily, hypothesis, regardless of whether hypothesis one or hypothesis two is true, you can, wait, what did Aaron say? Fluorescent ZPA is actually a good answer for this. What if you just fluorescently, what if you put a fluorescent marker into the ZPA and you see whether it's in the digits or whether it just like stays there? That would tell you what the ZPA is becoming. That would differentiate between hypothesis 1 and 2. Because if it's fluorescent, do I get 10 vowels for fluorescent? You can get 7. You can get 6 or 7, your choice. Anyways, but if you fluorescently label the ZPA and you see that digits 2, 3, 4 is also fluorescent later on in the life of the embryo, then you have to say that 1 is right. If you see that digits 2, 3, 4 are not fluorescent later on in the life of the embryo, that means hypothesis 2 is right. And that's a very simple way to check. You can ectopically transplant it somewhere else, but that's a test of necessity and sufficiency, which I will necessity and sufficiency is very important, and I will cover that, but that's not for today. And also, that will not tell you the answer to this question. So this is in developmental biology is called fate mapping. What you're basically doing is that you're putting in fluorescent markers into a particular cell or a group of cells, and you see how the fluorescent marker is distributed later on in development. For example, this green part is present mostly here. So you can say that this green part develops into this. This like pinkish magenta part is distributed like all over here. So you know what part of the embryo is formed by what type of cells, or what type of cells form what part of the embryo by fate mapping. Fate mapping, well, Chickwell chimera is another way for fate mapping, but I don't think we have enough time to cover it, so I won't cover it yet. So as the definition says, it's a diagram of an organism at specific stages of development indicating the fate of its component cells or regions. So it's the fate of its component cells. If the fate of the ZPA is to become digits 2, 3, 4, we would see it in the fate mapping, and hypothesis 1 would be right. If the fate of the ZPA is not to form digits 2, 3, 4, but to induce it using molecules, then we would see that the ZPA does not become digits 2, 3, 4. If you just remove the ZPA and then see limbs 1, 2, 3 form and not 2, 3, 4 within that eliminate hypothesis 1, which ZPA are you removing? If you remove ZPA, then no limbs will form at all. That's what will happen. If you remove the ZPA, won't anterior still form? If later on, we will discuss how the ZPA functions. But if you remove the ZPA, no limbs will form. Matter of fact, if you remove the ZPA, you don't even have an arm. You'll have something called meromelia. You'll have a slightly extended arm. And that's for the reason for this is very complicated, is because sonic hedgehog and FGF expression are engaged in a positive feedback loop. So if you don't, the FGF expression causes the proximal distal growth. Sonic hedgehog and FGF expression are in a positive feedback. If you don't have sonic hedgehog expression by the ZPA, That's how it functions, and we will see that later on. If you don't have sonic hedgehog function from the ZPA, then what you will see is that the FGFs do not function either, and you will have meromelia or very short, like you don't have an R. That's the point. Cut the ZPA in half. What do you mean cut the ZPA in half? I'll just move on, bro. That just doesn't work. We did this fate mapping, and we see FGF is fibroblast growth factor. There are lots of molecules in development, but yeah. Fate mapping of ZPA revealed that it only formed part of digit 4. If it only forms part of digit 4, that means part 2 and 3 has to be formed by something else. That means that the ZPA is actually inducing the formation of this structure. The ZPA does not physically form digits 2, 3, and 4. How do you read the results of fate mapping? I did not include the result of the fate mapping here because I was blocked. It was a very old article from 1975 that had it, but yeah, and I could not access it. That's why I don't have the fate mapping. But fate mapping would give you a fluorescent, it's like a immunohistochemistry. It will just fluorescently tell you where it is. Why are we cutting the ZPA, bro? Okay. Yes, you can try to explain in the chat, but yeah. Cutting the ZPA is not relevant. If you want to cut the apical ectodermal ridge, that's relevant. But we're not cutting the ZPA. For the purposes of this, we're not cutting the ZPA. So we found that hypothesis true is the correct hypothesis because the ZPA does not form all of these 3 digits. It only forms one of these. That means it induces the formation of digits 2 and 3. The hypothesis true is the correct one. It acts as an organizer that induces the formation of these 2 extra digits by some molecule. What is that molecule? That's what we're going to try and find out. A previous research also showed if you put a bead of retinoic acid in the anterior portion of the limb bud, and we have the ZPA here as we always do, we see duplications. They're not perfect. The reason they're not perfect is because Jaden, you don't have to be confused. All you need to do is just listen to me, and you won't be confused. Yes, the reason you see that, don't read the chat. Just listen. It'll be fine. You'll be fine. The reason you don't see perfect duplications is because you're already perturbing the embryo. That kind of messes up how it develops later on. That's why you don't see perfect duplication, but you see a very close to perfect duplication right here. This is normal, and when you put this, there's a ZPA at the posterior portion, and whenever you put a retinoic acid bead, RA bead, at the anterior portion, you see a duplication. This is very sus. So retinoic acid in the anterior portion leads to digit duplication. That means retinoic acid is somehow doing something that ZPA was doing in the anterior portion. So you may think ZPA induces the formation of it is. ZPA induces the formation of digits 2, 3, and 4 through some molecule. That molecule may be retinoic acid, since if we put retinoic acid, we have the formation of digits 2, 3, and 4. Later, we explored this hypothesis later on. So next, we looked at this molecule called sonic hedgehog. Sonic hedgehog, so this is something called fish, or not fish, but actually I forgot what this was called. I forgot whether this was a protein or mRNA, but I think it's in situ hybridization. So here we have the retinoic acid bead. This white arrow shows the retinoic acid bead. And this is definitely in situ hybridization. Yeah, this is not a gradient. Yeah, never mind. This is definitely in situ hybridization. So this is the expression of mRNAs. So we are looking at gene expression. So we see the black arrow. The black arrow shows us sonic hedgehog. So sonic hedgehog is here, right where ZPA should be. This is the posterior portion of the embryo, by the way. And this is the anterior portion of the embryo. The posterior portion of the embryo has a ZPA, and it also has sonic hedgehog. When you put in retinoic acid, we also see sonic hedgehog in the anterior portion. That's very interesting, and we see it here and here. This will later become the thumb. This will become the pinky. So we have this question. Is retinoic acid causing digit duplication directly by itself, or is it inducing the expression of sonic hedgehog? Sonic hedgehog is a gene and a corresponding protein for it. Or is it doing it indirectly? So one method is that sonic hedgehog is directly causing the duplication of the digits. The other method is that sonic hedgehog is inducing, you do not need CheapSig for this. And the second reason is that sonic hedgehog is inducing the expression of, no, it's not sonic hedgehog. Retinoic acid is inducing the expression of sonic hedgehog, and the sonic hedgehog is causing the digit duplication. So retinoic acid causes digit duplication. We know that because of this experiment. When we put retinoic acid bead and we saw digit duplication. Question is it doing it through sonic hedgehog or is it doing it directly? You can do that. You can do that. Yes, that is, you can do a knockout and that will give you the answer to that. Eastern blot, what was Eastern for? Was it carbohydrate moietase? Maybe. Anyways, I will keep moving. Post-translation, similar enough. Okay. So this was the picture I drew before. These are how the limb buds look. These are the wing buds, and these are the leg buds. We see in the posterior portion where the ZPs are supposed to be. This is a wild type embryo, by the way. In the posterior portion of each limb bud, we have the ZPA and we have the SHH, or sonic hedgehog. Now, we did another experiment. This was a very famous experiment. And so what we did is that we took some viruses. So someone proposed that we could do a knockout of SHH, but we could also do a knock-in experiment. Sometimes positive results are more appreciated than negative results in science because negative results can happen due to perturbations or problems that we can't expect. But positive results usually means something something has to happen for a positive result has to occur. And for a negative result, something has to be disrupted. And it's really easy to disrupt things, but it's really hard to make things happen. In this experiment, we're looking at a positive result. We are making viruses that are expressing sonic hedgehog. We are centrifuging the cells to make it into a pellet and a supernatant. The supernatant will have the viruses. Then we take the viruses and we inject it into the anterior portion of the limb bud in a resistant strain of host embryo. I will give 10 Baobucks to whoever can actually tell me why this has to be a resistant strain of host embryo and not a non-resistant strain. It has nothing to do with it dying. Resistant to the virus. So you're putting in viruses that express sonic hedgehog. And you're putting it in an embryo that is resistant to that virus. Why do you need resistant ones? It's not negative control type. It's not negative control. There's a very important reason. It's not as a control. It's not for control. I guess it is somewhat for control, but controlling what? What are we controlling by putting it in a resistant strain? It's because the virus will spread. We want the SHH to be specifically in the anterior portion. Whoever said it first gets 10 Baobucks. I don't know whoever said it. Cell spread. Okay, so whoever said it gets 10 Baobucks. Ajay, look into it. But yeah, Rachel gets 10 Baobucks. But yeah, if it's not a resistant strain, this virus will spread all over this place. And then sonic hedgehog is everywhere. You can't really say much. Because it's a resistant strain, then sonic hedgehog is localized only to this anterior portion. And then when you see this digit duplication, you can say that the presence of sonic hedgehog is what causes this digit duplication. And you can conclusively say that it is sonic. You can't conclusively say this, but you can probably say that the cell pellets have the viruses. The viruses go into the cell, and then it's it puts a sonic hedgehog DNA into the genome of the cell, and the cell expresses the viral genome, and then you have sonic hedgehog being expressed in those cells. So you can conclusively say, or I guess you can't conclusively say, but you can say sonic hedgehog is responsible for digit duplication. Later on in 2010, there was a paper in Science that more conclusively showed that it was sonic hedgehog and not retinoic acid that was causing digit duplication. And the ZPA was actually producing sonic hedgehog to produce extra digits. So during a normal embryo's development, the ZPA is in the posterior portion and it releases sonic hedgehog. The sonic hedgehog then causes the development of the posterior to anterior axis of our limb or of our like hands. So surely the fact that you had extra digits in the mice was because of a problem in sonic hedgehog. We know that much. RA induces a bunch of stuff. It induces a lot of Hogs genes and a lot of stuff. Can I get a Balbuck? Sure, you can get a Balbuck. Evan, you get one Balbuck. I'm very generous with Balbuck. If you want Balbuck, I'll give it to you. Whoever said I can get one, give him -1 Balbuck. Eric gets +2 because Eric is Eric. Anyways, we will keep moving. So sonic hedgehog is sufficient for the organization of digits and is usually expressed in the posterior region because ZPA expresses sonic hedgehog and ZPA is in the posterior region of the limb bud. Now that I think about it, maybe this was not the best lecture for first class because people are not listening to me, which is why everyone is confused. But like, that was too hard. Wait, this is generally not too hard. I am explaining everything in baby terms, bro. Wow, someone said this is not confusing, so we are good. This is much easier than anything on semis. If it's easier than anything on semis, why are you not a finalist? Like, bro, like, okay. We're just saying stuff to say stuff. Anyways, so this is Sonic Hedgehog. So when you see the Sonic Hedgehog there's a problem with Sonic Hedgehog, but there's no mutation in the Sonic Hedgehog gene. You look at the regulatory sequences. Maybe the promoter or the enhancer there's a problem with either of those. Accidentally, we found an enhancer for Sonic Hedgehog in a specific side of the embryo. You don't need to know limb R1 gene. You don't need to know the exons of it. We made transgenic mouse. So how are transgenic mice made? We put a gene, the gene inserts somewhere in the chromosome or the genome. And we saw that in one of the transgenic mice, we had digit duplications too. And we see where in the transgenic mice did the transgene insert. And we saw it insert at this one position 1,000 kilobase pairs away from Sonic Hedgehog. Now, we want to see whether this region is an enhancer. So, if the transgene inserted in an enhancer, then it might mess up the regulation of Sonic Hedgehog. That can cause its expression in both the anterior and the posterior portion. And as we saw in the virus experiment, if Sonic Hedgehog is expressed in the posterior portion, you'll get digit duplication. So we wanted to check whether this enhancer is actually an enhancer. And we made another transgenic mice. In this case, we have this enhancer and we have LAG-Z just next to it. LAG-Z is a gene that codes for a protein called beta-galactosidase. It's an enzyme that cleaves galactose. IPTG is uncleavable, but yes, IPTG is, yes, LAG-Z. So if you put galactose, am I muted? No, hopefully not. But if you put galactose, it is blue. That's how you do it. If you put the substrate for beta-galactosidase and you see that it turns blue in particular portions, then you can tell in those portions, beta-galactosidase is being expressed. In this case, we put in the substrate for beta-galactosidase and we saw that this reporter or this enhancer was expressing LAG-Z. So what do you need to know from this slide? From this slide, what we did first is that there was a mutation in like transgenic mice in which we saw digit duplication. We wanted to see where the transgene inserted because if it inserts into a gene inappropriately, it can disrupt it. We saw that it inserted at a particular region, and we wanted to see that whether the region was involved in regulating Sonic Hedgehog. That's why we checked whether it's an enhancer. And through other experiments, we saw that it is an enhancer. How do we check whether it's an enhancer? That's a technique that we will cover later. How do you conclusively say whether a portion of the gene is an enhancer? But in this case, we checked a portion of the genome that the transgene inserted in, and we said it's an enhancer. And then we made a model for how the mutation in the enhancer is causing the duplication of the digits. This is the enhancer. These are the activators. These activators act in the posterior region. This repressor acts in the anterior region. So in the posterior region, you would just have the activators, and they would activate sonic hedgehog expression. In the anterior region, however, you would have this repressor too, and this repressor would stop the expression of sonic hedgehog. However, whenever you have the transgene inserts in the repressor sequence, the sequence where the repressor binds. Bye, Kevin. And because of it, the repressor cannot bind to it in the posterior region. And because of that, in the posterior region, sonic hedgehog expression, not in the posterior region, the anterior region. Usually in the posterior region, sonic hedgehog is expressed by the ZPA. It is expressed because the activators bind to the enhancer and the repressor does not bind. In the anterior region, sonic hedgehog is not expressed because the repressor binds to sonic hedgehog enhancer, and that stops the expression of sonic hedgehog. But in this case, the transgene is inserting into the sequence that the repressor binds. And that's why the repressor cannot bind to the enhancer in the anterior portion of the limb bud. And that's why in the anterior portion of the limb bud, since the repressor cannot bind, there is no repression of sonic hedgehog expression. And so sonic hedgehog is expressed in both the anterior and the posterior portion, and that causes digit duplication. Now we have a pretty good idea of what may have happened in the hemimilic extratos, the mice that we saw on the first slide. It may be a problem with the enhancer. Later on, we did an experiment. We saw that there was a point mutation in the enhancer. This was a point mutation. This was a hemimilic extratose mutant. And the point mutation in the enhancer caused the expression. This is sonic hedgehog, by the way. The sonic hedgehog in this wild type is expressed in, this is the anterior, posterior, this is the anterior, this is posterior. Remember, this embryo is like bent. That's why anterior, posterior, you have to think of it like this. Anterior, posterior, and then it goes down to the anterior and posterior of the two limb buds. These are limb buds. These are not kidneys. Kidney cannot develop at this stage. A kidney is so much bigger than whatever this embryo is. But yes, in a wild type embryo, you have a correct enhancer sequence. And because of that, you have sonic hedgehog expression exclusively in the posterior region. However, in the mutant embryo, you have a mutation in the sonic hedgehog enhancer. Sonic hedgehog is fine, but because of the mutation in the enhancer, the repressor can no longer bind to it. And because of that, it is expressed in both the anterior and the posterior region. And because of that, we have digit duplication. And that's the entire lecture. I will shortly go over what we should take away from this. Remember, the purpose of this experiment was to look at what gene causes this mutation. What gene causes the digit duplication? We looked at a previous experiment where we saw digit duplications, and we saw it was because of a region of the embryo called the ZPA. We used fate mapping to prove that the ZPA induces the formation of the digits by secreting molecules. Then we looked at what molecules it secretes, and we saw retinoic acid might be the molecule. Then we looked at sonic hedgehog, and we see sonic hedgehog is induced by retinoic acid. And then we used viruses in order to say the sonic hedgehog is actually the one that causes the digit duplication, or sonic hedgehog is the one that directly causes the digit duplication, and the ZPA acts through sonic hedgehog. Then we looked at another case. In that case, we saw that there were digit duplications, and that was because of a transgene insertion. We looked at where it inserted, and we realized that place is an enhancer. And we made a model. We made a model that it inserted into a region of the enhancer where the repressor binds. So a repressor, if it binds to an enhancer, it can stop the expression. It can stop the activity of the enhancer. So if the activity of the enhancer is stopped, it won't be able to express the gene. So we proposed that the transgene is inserted into the repressor of the enhancer. And so the repressor is dysfunctional. So the repressor, since it's dysfunctional, can no longer repress enhancer in the anterior region, and we see SH expression in both the anterior and the posterior region of the limb bud. This entire thing is the enhancer. This is a part of the enhancer where the repressor binds. If this specific part is mutated by the insertion of a transgene, then the repressor cannot bind. That is what we are proposing is happening. The transgene is inserting into this specific green part. That's why the repressor can no longer bind to it. The repressor just goes away. Since the repressor goes away, in the anterior portion, we have no repression of sonic hedgehog expression. Since there is no repression of sonic hedgehog expression, sonic hedgehog is indeed expressed in the anterior portion. And later on, we see that sonic hedgehog in these HX mutants are expressed in both the anterior and the posterior portions. And that is because there is a mutation in the enhancer sequence. This is an example of how we can use forward genetics in order to find the gene that causes a particular phenotype. That is my entire presentation. Does anyone have any questions? I'm sure there's lots of questions. Jaden, please don't search of guns. You're a middle schooler. Am I teaching class on a cardboard? Yes, I am teaching class on a cardboard box. I was trying to buy Birkins for Kevin, but I'm too broke, so I'm teaching on a cardboard box. Exactly. I am homeless. Yes, thank you, Michael. Please donate to me. Instead of donating to, okay, I will stop recording first. Actually, Udeep, Ajay is here. Ajay, where are you? You're the co-host. You're stopping recording, bro. Bruh. Okay. I will stop recording.